Alright guys, in this video we're going to go over anaerobic respiration. Now, in the previous video, I went over aerobic respiration, where we have glucose that goes into glycolysis and spits out 2-pyruvate. Those 2-pyruvate will go through pyruvate decarboxylation, which produces acetyl-CoA. The acetyl-CoA will go into Krebs, and the NADH and the FADH2 will go through the electron transport chain. However, with anaerobic respiration, we have no oxygen. And with the lack of oxygen, we are cut off at this step right here. No other metabolic processes to create energy will go past glycolysis. Pyruvate decarboxylation, Krebs, and electron transport chain, they all need oxygen. So, anaerobes have no use for these things, but they still need to produce energy. And how do they do that? In order to produce more energy, microbes will have to keep doing glycolysis over and over again. However, glycolysis is a series of steps that uses several molecules over a period of time. And as we go through glycolysis, these precursors that we have used run out. So, molecules have to go through a process called fermentation. And what fermentation does is, it'll take the pyruvate that we have and it'll do one of two things. It'll convert it either to lactic acid or ethanol. Now I know I'm skipping a few steps here, but this isn't a biochemistry class, so don't concern yourselves with all the intermediate steps. These two things are undesirable for the microbe. They don't want lactic acid, they don't want ethanol. They produce no energy on their own. They're not like NADH or FADH2 where they're high energy yielding molecules. They don't want these, but what they do want, the thing that or the reason why they produce lactic acid, the reason why they produce ethanol is because, remember that we produce 2 NADH from glycolysis, in addition to the 2 ATP. Now let's focus on the NADH. In both of these processes, the NADH is converted back to NAD+. But why is this significant? It's significant because NAD+ is a key component to glycolysis. Without NAD+, glycolysis cannot go on. The main product that we want back is NAD+. So, the microbes will take NADH from glycolysis. They will then oxidize them and produce the NAD+. However, there is a consequence where they have to produce lactic acid or ethanol. But the main point here is that if, like in aerobic respiration, we want to get that 32 ATP, we have to do glycolysis 16 times because it only produces 2 ATP. So how do we do that? How do we do this 16 times? We do it by producing the ATP and the NADH. The NADH, as I said, will get oxidized back to NAD+. We'll take that NAD+, put it back into glycolysis so that we can do it over and over and over again. So, just to recap, for anaerobes, they do not go through aerobic respiration. They have to go through fermentation. And the point of fermentation is to take the NADH from glycolysis, oxidize it back to NAD+, and put it back into glycolysis. Because glycolysis does not need oxygen, but pyruvate decarboxylation, Krebs, and the electron transport chain, they all need oxygen. if we want to get this 32 ATP as we would out of aerobic respiration, we have to do glycolysis 16 times. That means we have to recycle the products from glycolysis 16 times. We have to recycle the NADH from glycolysis 16 times. I know it was a bit repetitive, but I just wanted to drive home the fact that these processes of converting pyruvate to ethanol and lactic acid produce a small amount of ATP. That'll probably be one of your test questions. But we don't get most of the energy from lactic acid and ethanol. It's pretty minute. The main thing that we want to do is do glycolysis again and again and again. If you guys have any questions about this, please feel free to shoot me an email, and I look forward to hearing from you guys.